What is the history of Alzheimer's genetics? Have you ever wondered how our understanding of Alzheimer's disease has evolved over time? The history of Alzheimer's genetics is a fascinating journey that began in the early 1900s. In 1906, Dr. Alois Alzheimer first described the disease, but it wasn't until the late 20th century that researchers began to focus on its genetic aspects. Initially, studies aimed to find biological markers of the disease. Researchers identified amyloid plaques and neurofibrillary tangles, which are abnormal structures found in the brains of people with Alzheimer's. These discoveries were crucial in understanding the disease's progression. A significant breakthrough occurred in the 1980s when scientists linked beta-amyloid protein to Alzheimer's. They found that this protein accumulates in plaques in the brains of affected individuals. This connection was particularly interesting because it related to a gene on chromosome 21. People with Down syndrome, who have an extra copy of this chromosome, often show signs of Alzheimer's pathology. As research continued into the early 1990s, scientists discovered specific gene mutations that increased beta-amyloid production. This led to the amyloid cascade hypothesis, which suggests that beta-amyloid accumulation triggers a series of events resulting in brain cell death. Key genes identified during this time included the amyloid precursor protein gene and presenilin genes 1 and 2. These genes are linked to rare inherited forms of early-onset Alzheimer's. In addition to amyloid research, scientists also began examining tau protein abnormalities. Tau usually helps stabilize structures in nerve cells, but in Alzheimer's, it becomes abnormal and forms tangles that disrupt cell function and lead to cell death. From around 2007 onward, advances in genome-wide association studies expanded the understanding of Alzheimer's genetics. Researchers began to identify common genetic variants that influence the risk of developing the disease. One of the most notable findings was the identification of the apolipoprotein E gene, particularly the apolipoprotein E epsilon for variant. This variant significantly increases the risk of late-onset Alzheimer's disease. The growing knowledge of genetics has practical applications for families concerned about hereditary health. Genetic testing can identify mutations in the amyloid precursor protein, presenilin 1, and presenilin 2 genes in families with early-onset Alzheimer's. This helps at-risk individuals understand their likelihood of developing the disease. Testing for apolipoprotein E variants can provide risk information but is not diagnostic on its own due to the complex nature of late-onset Alzheimer's. In summary, the history of Alzheimer's genetics shows a shift from identifying rare mutations to recognizing the complex interplay of multiple genes that influence risk. This understanding supports efforts to develop better diagnostic methods, targeted treatments, and personalized risk assessments for families worried about hereditary Alzheimer's disease.